Indeed, our favorite. Okay. So you guys actually need to be able to see something, huh? That'd be kind of nice. How about that? A little less dark. Yeah, right. Getting started. Promising. Okay. So chemical synapses, right? So it's kind of, we already talked a little bit about electrical, basically moving through gap junctions. Now we're talking about chemical synapses. Remember, I promise it's going to move quickly because basically an neuromuscular junction is a chemical synapse, right? So we already talked about the presynaptic terminal. We talked about that in the last chapter. We talked about the synaptic cleft. We talked about in the last chapter. We talked about the postsynaptic membrane. We talked about that in the last chapter, right? So all this stuff is in the last chapter. The only difference is the postsynaptic membrane in the last chapter was the sarcolemma. Now you can expand it from muscles to other neurons to gland cells. So the only difference here is you just basically explained your targets now. It's not just neuromuscular, it's neuroglandular or neural, I guess is what that would be, right? And so we already know that hanging out there at the synaptic terminal are your synaptic vesicles, which are signaled by calcium to release their neurotransmitters into the synaptic cleft so that you can communicate that action potential across to your target cells, right? And this should look very similar to you, yes? This is basically the exact same mechanism that we saw in the last chapter, only in this particular case, you're looking at a more broadened version of that. So you have your postsynaptic membrane, which we don't define as muscle now, but that's basically what it is. So ultimately, one of the pieces that's new to this, other than the basic mechanism, is this idea of synaptic. Here we go again. Synaptotagmin. This is kind of like the sensor that's associated with the synaptic vesicles that makes them sensitive to calcium. So a lot of times that's kind of like the lingering question in the neuromuscular junction discussion is, we know the calcium is a signal for these vesicles to release, but the question is, how do these vesicles know, right? We know how calcium triggers sarcomeric shortening, right, with its binding to troponin, but how does this actually work with this? Well, this synaptotagmin is basically the sensor for that. So this is kind of like the adapter, if you will, associated with these vesicles that allows them to be sensitive to calcium. So basically they have something similar. They basically just have a receptor for calcium that triggers them, and then they go and they release. So then basically once they release their neurotransmitter to the cleft. You can see then that triggers ligand-gated sodium channels to open up, and then from there, you basically are rolling through voltage-gated channels, right? So that's just all review. Now, what about neurotransmitter removal? We talked about this one too, didn't we? Remember acetylcholinesterase? Remember that one? So we talked a little bit about this one. So all neurotransmitters, we just talked about acetylcholine, but all neurotransmitters have to have some sort of a removal strategy, right? Because you don't want persistent signal left around in your cleft. And so that's true for all of these. They just have slightly different mechanisms for doing this. So we looked at the specific mechanism for acetylcholine removal, which was acetylcholinesterase. But oftentimes you either have to destroy it or you have to remove it. That's usually how you do it. Those are the two ways that you get lingering neurotransmitter out of the cleft. And remember, you got to do this quickly, right? Because you have to make way for the next action potential, which is oftentimes hard on the heels of the one that just came through. So you don't have a lot of time to reset your system in order to make you receptive to the next transmission. So here they basically just recapitulate the exact same acetylcholine mechanism that you saw in the last chapter, right? So you're releasing acetylcholine, and then it opens up the ligand gates. I didn't even touch it that time. Okay, so basically it binds to the ligand gates, which opens them up. And then of course you have your acetylcholine, which then gets torn apart by acetylcholinesterase into acetate, which you then excrete. And of course, choline, which you take back up. And then you combine that with an acetyl-CoA, which usually comes from metabolism. You lose the CoA, that delivers the acetate and smears that onto the choline. So you recapitulate your acetylcholine and load that into a vesicle. Okay. So the only difference here is you added a little bit of extra nuance there, in the sense that before we didn't tell you where that extra acetate was coming from to bind to the new choline that you just took up. It's actually coming from acetyl-CoA. For those of you who went through general biology, that may ring familiar to you. Remember acetyl-CoA? No, okay. So remember the acetylcholine is, acetyl-CoA is the starting material for the Krebs cycle and metabolism. So you have a lot of it. It's also a glucose derivative, right? So you get to acetylcholine by going through glycolysis and by getting going through pyruvate oxidation, and that's what generates your acetyl-CoA. So you actually have to do a little bit of glucose breakdown, a little bit of metabolism. You go about halfway through metabolism in order to get to a hold of your acetyl-CoA. And so that's important, right? Because we always know that it takes a lot of glucose in order to do heavy concentration. This is part of the reason why. Not only are you supplying nutrients with glucose, but you're also supplying acetyl-CoA for your neurotransmitter connection. Now, what about removal of different, like norepinephrine, right? A different neurotransmitter. So we understand acetyl-CoA, but now what about norepinephrine? So norepinephrine has a very similar sort of a scenario, right? So basically you get synaptic release, your norepinephrine goes into the cleft just like before, binds to its receptor, has whatever response it has, and then it releases, right? So you have to have your receptor release of norepinephrine, and then norepinephrine just gets taken up. So you don't metabolize it, you don't cleave it, you don't chop it up. You don't want to mess with norepinephrine. You just want to take it back up. And then what you do is you essentially just reload that into a vesicle, or you convert that into some sort of a waste metabolite. Depends on what you need, right? So if you've got a heavy norepinephrine response right now, then you probably want to repackage that into some vesicles so that you can re-stimulate. Okay, so it's more of a cycle in this case, and it's more associated with just the uptake piece of it. So the same way the acetylcholine, the choline component is uptaken by these transporters, the norepinephrine is just taken up by the transporters. Now, in terms of number 4, like when you have these inactive metabolites, what does this one look like? So this is basically broken up by an enzyme called monoamine oxidase, MAO, which breaks down the norepinephrine into these waste molecules. And the reason why this is important is because MAO, actually MAO deficiency, is actually a genetic disorder that actually creates excessive aggressiveness in individuals. It's actually a genetic disorder that really has a very strong negative behavioral effect. And we're talking like MAO deficient individuals, like being prone to bouts of aggression where like a forklift operator will just like out of the blue, just drop the load on their boss or something like that. I mean, just like out of the blue, like they're just like random acts of violence. And they're known for that. These, MAO deficients, like, you're like, that's me. It's like I've gone through a few bosses that way. Right. So so it was just like an overall, like just a really snap kind of a thing. And so some of that is MAO, not all of it, by the way. We see a lot more of that now. So there's obviously a lot of that that's controlled. So there's people who will do that now that they're just a-holes. And, not everybody's MAO. MAO is actually a rare genetic disorder. I would argue most people who are probably killing their boss probably should go to jail because you don't have an excuse for that one. But go ahead.
Okay, so how does that fit in? So like, is that something that you're born with? Or is that something
For that one in particular, the one that we originally identified as MAO deficiency is an actual defect in the gene that encodes monoamine oxidase. So basically what that means is this process here is deficient. So you don't have a good way of breaking down the norepinephrine, which is an adrenaline. And so you don't have a really good way of backing off your adrenaline rush. And so since the adrenaline is your neurotransmitter behind your fight or flight, then you're equally likely to fight as flight. So if you're not able to back off of that adrenaline rush, then you kind of have this perpetual cycle going on. And you're sort of stuck in an adrenaline loop. What?
How does that work with PTSD? Because a lot of people with PTSD have that, just no hidden switch on, switch off?
So the stress cycles, so this is heavily related to all those stress cycles because stress cycles like that, anxiety disorders, all that sort of stuff is all driven by the same group of neurotransmitters because the adrenalins, norepinephrine and epinephrine, are the primary neurotransmitters behind your fight or flight response. So generally speaking, all of those stress disorders, induced or otherwise, PTSD would be an example of an induced stress disorder, because the idea is that it's post-traumatic, not pre-traumatic, right? So a stress disorder pre-traumatic would be a different thing, right? So whether it's induced or not, it means that there's some sort of a compromise going on in here, either with this specifically or with the interpretation of it, right? Because there's a lot of interpretation, like threat interpretation, that is also factored into your threat response and your stress response associated with it. So it's not just this easy. There's also a very convoluted interpretational component of it with your brain, right? The idea that, you really strongly feel threat where there's no threat, right? So that kind of an idea, that's an example of a misinterpretation of the environment, which is not necessarily a neurotransmitter thing as much as it is an interpretational thing that's going on in the brain. So it's an abnormal interpretation of the circumstances that's then triggering this, and then you start to get this sort of fight or flight thing going on. So that's a fascinating field, though. Actually, it's kind of a neuropsychology kind of a field where both neuro and psychology are kind of dovetailing together because it's a behavioral spectrum along with a very strong neurological spectrum. So it's a fusion of the two. So it's pretty new. I mean, we had almost zero, to offer in this field for probably until very recently. And we're still not really quite sure how it all works. So it's a lot of work is going into that. The general mental health is going along with it, but the stress-induced disorders are probably the more acute version of the overall pantheon of mental health areas. What?
Where does the anger and the surge come from?
Um,
If it's a threat, it's fight or flight. What causes the anger and surge that comes with that?
that'd be your system. That would be your internal. So like, for instance, oftentimes with like stress disorders, what you'll find is that there's triggers that will throw you into, and sometimes the triggers make sense and sometimes they don't. Because it doesn't really matter what it is, right? I mean, it's, somebody doesn't have to be carrying a gun for you to trigger. It could be something that you wouldn't even expect to be a trigger, and it just triggers it. It's not about necessarily the physiology or the trigger itself. It's about how the brain is processing that particular event. And that's the reason why it's a disorder, why it's a stress disorder, is because a lot of times the brain is misinterpreting that event, right? So it's like, this is not a threat, so this should not be a trigger. But for some reason, there's something in the brain that's processing this as a threat. And then once you do that, then you just turn it over to your fight or flight response, and it's all visceral response at that point. That's where the surge comes into play. So, when you take a look at neurotransmitters, the important thing about these guys is essentially they have their own receptor, right? So you can't basically have a neurotransmitter or one receptor that fits all neurotransmitters. So there's an acetylcholine receptor for the acetylcholine. There's a norepinephrine receptor for the epinephrine, right? So if you want to be responsive to a neurotransmitter, then you have to build that receptor into your target site in order to be responsive to that one. Not only that, but when you have a neurotransmitter, that goes two ways, right? The neurotransmitter will only respond to its receptor and to the cells that are carrying that. So if you have a target site and you've got a bunch of norepinephrine in your cleft, but you don't have any norepinephrine receptors, guess what? You're not going to get a response. It's a two-way process. You have to have the neurotransmitter and you have to have the receptor. If you miss any of those, then essentially you're dead to that response. Now, neurotransmitters in general fall in two different classes, right? We usually think of excitatory neurotransmitters, like norepinephrine, for instance, is an excitatory one, so is acetylcholine. But some are inhibitory in nature. Like, for instance, the transmitter GABA is typically an inhibitor. And remember, we said that one way, not the only way, but one way that neurotransmitters can create inhibition in action potential response is by We mentioned it. By hyperpolarization. I don't know how to spell that. So pretend that's hyperpolarization. Or rather, I should say, I don't want to take the time to try to spell it correctly, right? Remember we said that? We said that one of the ways that you can create immunity to impulse is by hyperpolarizing the membrane, and that kind of inoculates you. So that's kind of an inhibitory sort of a thing. So you're less likely to excite that membrane at that particular point. So that's one way that you can do that. So a couple of neurotransmitters, norepinephrine is an example of these, can actually modulate its own release by binding to the presynaptic terminal. This is kind of an interesting little wrinkle, right? So this basically gives you self-regulation. So not only can it regulate the response of the target cell, but it can also regulate its own production or own release rather, by having its own receptors on the presynaptic side of things, modulating the release of it, right? So for instance, calcium is triggering the release of it. What if you wanted to buffer that a little bit, slow that response down a little bit? Well, then you can add some of your receptors into the presynaptic terminal, and then by binding to that, it can have the effect of muting that release signal, right? So you can kind of attenuate it. It's almost like a dimmer switch, right? Modulation, typically, whenever you see the term modulation, it means it acts like a dimmer switch. And that gives you some nice control over the overall effect, right? Which is kind of important because, I mean, obviously you don't want to fight or run at the wrong times or inappropriate times. And you don't want to have the exact same response, right? So you don't have the, you know, the absolute bone melting life or death stress response that you have in one scenario for something that's considerably lower threat, right? So you don't want to have the drop dead stress response when all you see is a spider on the ground. That's a little overkill, right? So you want the ability to attenuate that. You can attenuate it by attenuating the release of the norepinephrine, which would attenuate your stress response. Those are all things. So this is basically kind of the catch-all. So you can see here, you got your graded potential, which then rises to threshold, at which point you get depolarization. And then that starts to roll continuously down the axon. The segment that will repolarize, hyperpolarize, and then basically recalibrate. And then ultimately, if you are myelinated, you'll move by saltatory conduction. Remember, that was the jumping one from jumping from node to node to node, until eventually you get down to your synaptic terminus, where the calcium release will bind to synaptotagmin and then cause the vesicles to release their neurotransmitter into the cleft, which will then bind to the receptor in the target cell, and then that will elicit whatever that response is, right? In the muscles, that response is to send it for the purpose of contraction. In glands, it's for the purpose of secretion, and things of that nature. So it's whatever your target is designed to do. Once you communicate that message across to your target cell, then it will basically communicate that message to the other cell. And then, of course, you got to remove everything out, right? So then once you remove everything out, then you reset the system, and then you have yourself ready for another wave coming. Most people like a nice little list. This is it. You've got your graded potential starting off, right? Which basically depolarizes. Typically, this is happening at the axon hillock, right? That's that little swelling at the cell body side of the axon. This will trip those initial gates open, which will create this sort of rolling depolarization. And then, of course, after depolarization rolls through, you'll do repolarization. And then you'll propagate that either continuously or in a saltatory fashion, depending on whether or not you're just like rolling from one segment of the membrane to the next, or if you're jumping from node to node, depends on whether or not you're myelinated or unmyelinated. Then you'll reach to the synaptic junction where you will release your neurotransmitters into the cleft, and then they will communicate with your target cell. Okay, so that's kind of the wrap up, essentially. So kind of put that into a nice little list so that you guys can sort of roll through that stepwise progression. It's actually quite functional. By the way, the axon hillock where this original depolarization is happening is in this area right here. So this is your axon hillock. Is that kind of little open area right there. That's where it starts. And then it starts to roll through the axon until it reaches the terminus. Okay. So what about neurotransmitters and neuromodulators? Well, obviously they're chemical messengers, right? And so we have a lot of different types of neurotransmitters. We just talked about 2 so far. There's actually at least 100, especially if you toss in the neuromodulators. Remember, these basically are sort of secondary to neurotransmitters. And these are the dimmer switches. So the response is already there. What you're doing is you're just making it either more aggressive, or you're kind of muting that response and sort of softening it a little bit. Now, what if you wanted to graduate and become a neurotransmitter someday? Then what would you need? What's the criteria that you would need to have to meet? Well, first of all, it needs to act like a neurotransmitter, right? First of all, the neuron needs to make it. Nobody else, the neuron. And then it has to be stored within synaptic vesicles, just like acetylcholine. And that's going to be found at those presynaptic terminals. Okay. So if you're going to be a neurotransmitter, you got to act like a neurotransmitter, made by the neuron, stored in synaptic vesicles in that presynaptic terminal. So what's the other thing? The other thing is that it has to be released into the synaptic cleft by the same exact mechanism that neurotransmitters are released. That is to say, your action potential will stimulate the release of these vesicles to release you into the synaptic cleft by exocytosis. Okay, so that's number 2, criteria number 2 that you have to meet. What's the other thing that neurotransmitters do? Well, they bind to a specific receptor in your postsynaptic membrane. So you have to do that. You have to have like an acetylcholine receptor that's maybe part of a ligand gate. That's okay. But you have to have some sort of a receptor that binds to you on the other side. And then what else do you have to do? You have to have some sort of a response. So if you bind to everything and nothing happens, then you're not really much of a neurotransmitter, are you? So the idea is there has to be some sort of a response in that postsynaptic cell. That is to say, you had to have told that cell to do something. Now, notice what we didn't say. We didn't say it has to do something positive. It could potentially do something negative, right? A legitimate response is to say, don't do this. That would be an inhibitory neurotransmitter, right? So that would be an example of one. So how do you classify these guys? Well, you can classify them in a couple of ways. Either according to their structure, which we do oftentimes. You can classify them in their effect, like for instance, excitatory or inhibitory in that nature. Or in the way that they actually interact with their targets. Okay, so those are 3 major ways to look at it. So when you take a look at their activity, whether they're excitatory or inhibitory, there's a couple of ways that they can manage to do this, right? So first of all there's what's called ionotropic neurotransmitters. These are the ones we talked about mostly. These are the ones that are binding to ion channels and they're opening the ion channel. This is the one we thought about and we kind of sketched out in the neuromuscular junction talk, right? So that first ligand gate of binding acetylcholine, that would be an ionotropic effect. It has an effect on an ion channel. So the other one, which would be a metabotropic effect, is essentially a different strategy. It's coupled to a type of receptor. It's called a G protein receptor. And I don't want to get into this a whole lot, but basically when you take a look at G proteins, and this gets into signal transduction and it gets deep into signal transduction. But basically what G proteins are is you have a membrane and then there's a receptor out there, and it's basically tethered to a series of proteins on the inside. So when your neurotransmitter binds to your receptor, it activates this, what's called G protein, which will then activate several players in its cascade, which eventually will communicate with the cell inside. Okay, so there's multiple different patterns of cell signaling. And most of them involve this kind of relay system. The message is received on the outside by the receptor. That message is then taken by the receptor and relayed to a cascade of proteins inside the cell, which will ultimately terminate into the ultimate effector. That is to say, the protein who does whatever the work is that needs to be done. But there is it's like a grapevine effect, right? There is a long cascade of one person giving the message to another, pass it on to the other, pass it on to the other, pass it on to the other, pass it on to the other. And then finally, the person who needs the instruction finally gets it, okay? There's multiple patterns of that. G protein coupling is a type of that pattern. Starting with what's called a G protein at the periphery that takes that message and passes it on to the cascade. So it's different than what we saw with acetylcholine opening up a gate, which would be ionotropic. Okay. So that's an example of their mechanism of action, like how they communicate this to their target. So what about chemical classification? We said we could do that one. This is probably the one we do, actually. So we can categorize them in terms of their basic structural, their chemical structure. Obviously, acetylcholine is the gram poobah, right? That's the one we understand the best. It's acetate attached to choline, so it's a fairly simple structure. That's the one we know the best. But some of these, that's what's called a catecholamine. But some of these basically are essentially what's called biogenic amines, which is an organic chemistry group. You don't have to worry about that, right? So we have catecholamines and indolamines. For instance, serotonin, which is kind of associated with mood and stuff like that. Dopamine, which is a really common neurotransmitter, which is involved in movement, tactic movements as well as learning as well. So dopamine is one of these. Norepinephrine is another one of these types. So you kind of structure them together as biogenic amines. The other one, some of them are actually amino acids, right? So for instance, I mentioned GABA before, which is an inhibitory type of neurotransmitter. That's actually glutamic something of butyric acid or something like that. So it's glutamate derivative. So it's actually an amino acid. Glycine can act as a neurotransmitter, as can glutamate. So there's a couple of amino acids that can actually act as neurotransmitters. Some purines. Where did you hear about purines before? Purines are actually the type, one half of the types of DNA bases, right? Adenine and guanine are types of purines. So these are nitrogenous bases that are typically coming from nucleic acids. ATP is a type of purine. And so some of these purines can actually act as neurotransmitters or neuromodulators. What about neuropeptides? So neuropeptides basically means a protein or a peptide, a short chunk of protein with neuro ability. That is kind of neurocommunicative ability, right? So you have short little chains, chunks of amino acids that will then be communicative. Like for instance, endorphins, right, which is kind of like the body's natural morphine, is a neuropeptide. Substance P, which is involved in digestion, is also a neuropeptide. So very short chunks of amino acids, probably too small to even be considered proteins. So they're just like little peptides, so like little itty bitty pieces, almost look like little shreds, scraps of leftover degraded protein, almost like if you didn't know better. But these actually turn out to be neuropeptides. And then some gases and lipids are actually can act as neurotransmitters. Good example of a gas is nitric oxide, NO. That can actually act as a neurotransmitter. Carbon monoxide can act as a neurotransmitter, an inhibitory neurotransmitter. Hence the reason why carbon monoxide is poisonous to us, because it basically shuts down all of our synaptic clefts, and that's how we die, essentially. Endocannabinoids, right? Basically, these are lipid-derived versions. These can also be neurotransmitters. Of course, cannabis, cannabinoid, right? So our favorite plant, Colorado's cash crop, right? Right. I'm just saying, it is probably quickly becoming Colorado's cash crop. Actually, that's probably no joke. I mean, it's outpowering wheat and anything else out there. I mean,
The strongest substance on the market right now is hemp.
really? Wow.
Ben say it's okay.
Yep.
You find 8, the 9 and 10, delta 8, 9 and 10.
Yeah.
And they have structured them together. Now they've come up with like a 35% THC derivative of CBD, which is like really screwing people up.
Yeah. Yeah, because hemp is supposed to be the safe one that you can go to and not have to worry about all the restrictions of CBD and stuff like that.
It's supposed to be below 0.3.
Yeah.
And a lot of it's coming up around 7 and 10%. And what they're doing is they're
Yeah. Using those two.
they're breaking it all the way down until they come up with a substance that is a microtoxin or
Yeah.
in tractor trailer drivers and people who stop at the pit stops and the fuel stops are picking up CBD and then what they're getting and all this stuff is causing havoc across the country.
Yeah.
They're all like, why do we feel sick?
Yeah.
What's going on? We have a package. Oh, we're just doing it all.
Yeah.
So, yeah, it's actually dangerous.
It is
It should be
dangerous.
very
That's why
clear about what it is.
it's like hemp isn't sold in Colorado. It's sold outside Colorado, like you can't get any of that stuff from the inside. And that's why cannabis, but they're changing the laws. They just came out with a whole bunch of laws yesterday. They changed a bunch of stuff for the hemp growers.
Law has
So,
Yeah. Yeah. Marijuana regulation is still Wild West territory. Yeah. So it's
well, even where- this is
it's
the
more
best-
wild than law.
Colorado has the best system in the country right now compared to other states.
That makes sense though, because we were one of the first ones.
We're the first ones.
Yeah. So we've had a little while to figure this out the hard way. But for you table people, here's your table. Right. Just kind of puts them all together. Right. You have your acetylcholine and your mechanism of action. So it actually kind of breaks it all down for you. So you can kind of categorize these and kind of organize these really nicely. Your biogenic amines, which are really cool, your amino acids. And then of course your purines, which is adenosine largely for the most part. Your neuropeptides and your gases and lipids. So these are, this is kind of like basically what you went through, only it's kind of all nice and on a nice little chart for you. Okay, this is just purely your study piece. Okay. I know what you're thinking. You're thinking, hey, we get to leave early. No, you don't.
Where did all of that information?
Yeah. The brain has limited. It's like you gotta upgrade your memory to Terra or to multiple Terra. What's after Terra anyway? Quanta? Yeah.
Gigabytes.
What is it?
Gigabytes.
Tiga?
Giga. It's 100 terabytes. Or 1,000 terabytes.
Yeah, you're like, yeah, that.
Yeah.
Okay, is it gonna happen today? Okay. So a couple of things I want to yes. What is this? Wi-Fi, right? Dirt to pigs. Yeah. Let's log on to that one. Sorry, I'm babbling. Just ignore me for the next couple of minutes. It's not really what I wanted, though, is it? This has been doing this to me like all day long. And sometimes it's Zoom. And sometimes it's the Wi-Fi. There we go. Okay. Let's take a look at okay, so now we're going to go through cherry picking. Sorry for the folks online. I was basically doing that entire, I was just basically pulling up the PowerPoint for the new chapter, which is chapter 12. Yeah. I know. They're all like, I knew that schmuck was a flake. Okay. Referring to me. That's okay. I'm used to it. So let's go ahead and cherry pick, right? So this is the cherry picking. Couple of things I want to cover first, right? Just basic spinal cord. Don't want to really spend a whole lot of time on the spinal nerves, right? So I may come back to this chapter just to sort of fill in a couple of pieces in here as we have time, but I want to hit the first parts first, and that is spinal cord anatomy. This will actually go quite well along with your studies in lab. Okay, so first of all, this is basically your spinal cord. So you can see it's basically going to be tucked into your vertebra, and you can see what it actually looks like. So the first thing you're going to notice is you have what's called the epidural space, epi upon the dura. So like if you get an epidural, this is basically where they're injecting it, is right into that area, into that epidural space, this space right here. Right. So as you kind of zoom in, then you can actually see that as you move inward, you actually have your meninges. Your meninges are the coverings of your central nervous system. It's around your spinal cord. It's also around your brain. The outermost layer is your dura mater. So that's kind of a tough connective tissue layer. That's the little pink layer. Then below that is what's called the subdural space. So they're trying to actually point to this little dark line right there. That's the subdural space. Like a subdural hematoma is pooling of blood in that area. That's usually if you've taken a pretty good smack to the head. Okay. and you ruptured blood vessels and you got pooling blood in there. That's a bad idea because it builds up pressure and there's no way to relieve that pressure because you're talking about a small confined space encased in hard bone, right? Which is the reason why hematomas are bad. As you move in, the next layer in, the middle layer, is called the arachnoid mater. So the arachnoid mater is this little yellow golden area right in here. And it kind of has a lacy, cobwebby look to it. And that's why it's called arachnoid. Arachnids are spiders. And then as you start to move in, there's another space. This is the subarachnoid space. This is typically where you have a lot of cerebral spinal fluid circulating around in that particular area, in that particular space. And then right on top of the actual nervous tissue itself is going to be the last of your three meningeal layers, the pia mater, which looks like a little layer of cellophane resting right on top of the actual spinal cord itself. Now, then you're also going to notice that in the spinal cord, you have this basically this kind of long cord area. And we're going to take a look at this in specific. But attached to this are going to be two different roots. So this one, the dorsal root, so this is your back up here, right? This is your front, that's your chest, right? So you have your dorsal root coming out of the dorsal side, and then on the ventral side, you have your ventral root. Now the dorsal root has a little bit of a swelling right here, which you guys saw on the spinal cord model. That's the dorsal root ganglion. Remember, ganglion is a collection of cell bodies. So this is a major a transfer station, if you will, for messages coming and going into the spinal cord and out of the spinal cord. And then the two will fuse, and when they fuse together, then they'll create what's called the spinal nerve. Now, once you're the spinal nerve, you exit out of the vertebra, and then you start to divide up into typically a bunch of brachial plexuses. So a lot of like nerves that sort of, there. So you start to break out into all of these nerves that start to wrap around the body and sort of innervate all the body, right? These are called plexuses. You got a cervical plexus, you got a brachial plexus, you got a thoracic plexus, a lumbar plexus. So these are just like little areas where all these spinal nerves are coming out, and then they're sort of they're fusing and splitting and fusing and splitting, and so there's all sorts of stuff going on to basically create all of those peripheral nerves that you essentially associate with your body. But what I want to do is I want to take a look at the cross section of the spinal cord itself, which is this guy right here. So here is our dorsal root. So you can see we have our dorsal root here, our ventral root here. There's our dorsal root ganglion. Now in the actual spinal cord, it's broken up into gray matter and white matter. The white matter is going to be broken up into columns. So for instance, this area right here is going to be what's called the dorsal or posterior column. Why? Because this here is your back. It's the dorsal region. Then this area down here of white matter is what's called your ventral or anterior column. And then you have lateral columns on the side of the butterfly. Okay, so those are your lateral columns. That's how your white matter is broken up. Then the butterfly itself is the gray matter. Again, remember, the gray matter is a collection of cell bodies. The white matter is a collection of axons. So these are axonal tracts in these columns. So the gray matter is broken up into what we call horns, right? So first of all, the big horn up here is what's called the dorsal or posterior horn. Then on the bottom here, you have the ventral or anterior horn. And then in the middle, right about here, is what's called your lateral horn. Notice it's the exact same organization. You have posterior, anterior, and lateral columns, posterior, anterior, and lateral horns, right? One is talking about the white matter, the other one's talking about the gray matter. And so this is kind of what it looks like in cross section. So a lot of you guys kind of saw pictures of this and you've seen this. Now, this big groove right here is kind of like a major landmark. This is what's called your anterior median fissure. That kind of tells you where the anterior region is. The little line, excuse me, I lied. The little, where's a good image of it? If you take a look at your your anterior median fissure, it doesn't go all the way to the gray matter. There's like a little bridge right there, white matter. That one is what's called the white commissure. And then you can see this little stretch right here of gray matter is the gray commissure. And then in the middle is what's called your central canal, and your central canal is full of cerebral spinal fluid that ultimately comes from your brain. So it goes through that fourth ventricle in your brain and then basically into the spinal cord central canal. Okay. So that's the organization of your spinal cord. Now, when you're taking a look at communication, one of the things you'll notice is that the white matter, right, because these are tracks. Remember we said in the peripheral system, tracks are bundles of axons. Remember, we said nerves are bundles of axons in the central nervous system. Nerve tracks are bundles of axons the other way around. Nerve tracks are bundles of axons in the central nervous system, and nerves are bundles of axons in the peripheral system, right? So what we have, remember, is ascending and descending nerve tracks here in your spinal cord. Notice it's all in the white matter. Why? Because nerve tracks are myelinated. That's why they're white. They've got that myelin sheath around them because they're moving signals all up and down the spinal cord. But remember, we have two divisions of our nervous system, right? We have the afferent, the arrival, which is taking sensory information and sending it up to the brain, and the efferent, right, which is exiting, which is taking responsive information, that is your decision that you made, and it's sending it down the spinal cord to your targets, so your targets know what to do. What that means is in your spinal cord, you have both ascending tracks, which are part of the afferent system, and descending tracks coming from the brain, which are the efferent system. And this is how they're organized. So you can see in the columns, right, in the dorsal or posterior column, you have some major ascending tracks here, so in green, right? And then a lot in the lateral columns as well. And then you have a little bit, mostly in the anterior column, it's mostly going to be descending. So that's your response system coming down to your descending side. You can see that how you have your communication tracks sort of peppered throughout the white matter of the columns themselves. So that's a little bit of structure for your spinal cord. Now what you want to do is want to take a look at communication. So how does this actually work? So here's kind of how some of the nerves work in response to the actual roots themselves. So we've got our spinal cord. We already know we have our columns there. We already know we have our ascending and descending tracks in the white matter. But what basically happens? So ultimately what's happening is in the dorsal root, notice you have spinal nerves, you have information coming in that then gets processed in the dorsal root ganglion, but this is sensory information coming into your spinal cord. And so this sensory information will then go into that posterior horn. Typically, it will get relayed oftentimes with what's called an interneuron, where we said there's three different types of interneurons, right? There's afferent, efferent, and interneurons. Interneurons are just basically passing it from one neuron to the next. That's what the interneuron is doing. And here you can see it's synapsing with an outgoing neuron that's basically heading into the ventral root. And the ventral root typically is where you have your somatic motor neurons. So what does that mean? Ventral root is basically motor and efferent in nature. The dorsal root is oftentimes sensory and afferent in nature. So what does it mean? That means the sensory stimulus you're getting from your body, that you're collecting from your body, is coming in through the dorsal root, processed through the dorsal root ganglion, and going into your spinal cord. And then oftentimes what'll happen is it'll go through the gray matter, right, the horns, and then it'll get relayed to the anterior side, and then the response, the efferent response, that outgoing response, will go through the ventral root, and then you can see that this basically goes out to your target. In this case, it's a motor neuron, so you're looking at musculature, okay? So there's two different ways you can do this. You can either do it with or without a neuron. So the difference between purple and blue is simply with or without an interneuron. The purple one, you basically relayed through an interneuron between your sensory and your motor. The blue one, however, was direct synapse between sensory and motor. So you didn't have an interneuron there. Is there a wrong way to do it? No. Do you always need an interneuron? No. Right? So you can have both. You can have direct transmission or you can have interneuron transmission. Okay. It just depends on the nature of how you're communicating. But this is the general gist, the general basic communication pattern that you see coming into the spinal cord. That make sense? It's like, that's okay. The green arrows going off to the side. These guys? So this goes up to the brain, right? So the information, the sensory information comes in, communicates to the spinal nerve. There are some immediate things that the spinal cord can actually process and deal with. But what happens is through these ascending tracks, that message also goes straight up to the brain. So your entire central nervous system is getting this sensory input. Your spinal cord is processing it, and your brain is getting a copy of it. Okay. Now, it doesn't necessarily mean that your spinal cord acts on it, but it can if it wants to. Okay. Generally speaking, what usually happens is the sensory information will come through the dorsal root, then it'll get processed in the spinal cord. That'll get sent up through the ascending tracks through the spinal cord up to the brain, and it'll get dispersed to the correct portion of the brain to deal with it. Then a decision will be made, and then the response will come down through the efferent side of things, the descending tracks of the spinal cord, and then it'll arrive at the spinal cord and will exit out the ventral root en route to the target. Yep. So you send everything up to the brain makes a decision about it, sends out what it wants to do about it, and sends that down through the spinal cord, through the descending tracks for that particular action to occur. Now, what happens when you have a reflex? Well, a reflex basically, and there's a lot of different reflexes. I don't want to get too much into them because I want to stay targeted and focused. But here's basically how a reflex works. A reflex is something where you have, it's kind of like a short circuit from normal transmission. That only goes to the spinal cord. Remember, most of your sensory information is coming in and going to the brain. The brain makes a decision and sends it out through the efferent descending tracks. And then that's the message that goes out the ventral root. However, in a reflex arc, it's a short circuit of that process. So generally what happens is you get some sort of sensation. It's coming through the dorsal side. That's still normal. But then what happens is it's going to basically go through an interneuron. It's going to go immediately right back out through the ventral in a motor response. Notice what's happened is you short-circuited. You didn't wait for the brain's response at this one. You immediately went through. So that interneuron setup is what we see predominantly when you look at reflex arcs. And there's multiple different types of reflexes. They're almost always survival in nature, right? So typically, here's a good example. If you put your hand on a red hot stove, do you have to think about that one before you take it off? Before you even know it's happening, right? Your hand is already off. Did you even think about it? You don't even realize half the time what happened until after the fact. That's a reflex, right? What happened was the sensory information went straight to your spinal cord and triggered the reflex, which went straight back out to your hand saying, okay, we don't even need the brain on this one. Get it off, right? And then what you do is you CC the brain just as an FYI. FYI, you just burned your hand, fool. Go get yourself taken care of, right? The fool, that was my part. I just added that, right? It's not like, but that's kind of what it is, right? Because most of your reflexes are survival in nature, right? So if I like went up to somebody and they'd punch me, but if I went up to somebody and without her knowing it, I went like that. What does she automatically do? Look at her eyes,
Pull back and twist.
right? Her eyes naturally blink. Did she think about blinking? It was reflexive, right? So that's a survival reflex. You didn't have to think about that one, right? That was basically your reflex saying, close your eyes because there's some danger coming to your eyes. You don't want something to get into your eyes. It's an automatic reflex, right? Another good thing with the eyes, I'm picking like because you're close to me. But this is what you get, right? Being this on this side of the now, of course, next week she's gonna be on that side of the room. But anyway, that's okay. I'll just go over there then. Right? Is that okay? I'll just follow you around the room. Right? So, okay, anyway. Right. The other one is pupil dilation. You've done this one a lot, right? Heard about this one? Your doctor kind of shines a light in your eyes. What are they trying to look at? Your stunning blue eyes. If you have blue eyes, right? In your case, it's like, I don't have blue eyes, so it's something else that's stunning in there, right? All right. Right. So that's, I mean, is that what they're looking for? No. What are they looking at? They're looking at pupil dilation, right? Which is reflexive, because the idea is when you shine the light in there, what happens to your pupils? Right? They automatically constrict. Why? Because they are shutting down the amount of light so that you don't damage your retina. Same thing is also true when you get into a dark room. Immediately, what do your eyes do? They dilate, so they let in more light so that you can see better. Well, here's the thing. If you have trauma, head trauma or something like that, that is that's what they're doing. They're trying to determine, like you said, brain damage, because what that means is if they see dilation in one eye but not the other, bilateral asymmetry, then that suggests that there is some brain damage on one side of the brain, right? Because that brain reflex is not being controlled properly. You're shining the light in there and it's not constricting. Yeah, because it's like, it's because of the optic chiasma. So if they shine in the left, it's telling them there's right brain damage associated with that. If it's bilaterally symmetrical, non-dilation or non-responsive, that's a very bad sign, right? Because that's usually, like, especially a first responder, that's usually their first sign that there's problems, and like serious problems, right? Because that means that the brain has been compromised to the effect where it can't even manage these reflexes, which are very, very visceral. That is a very bad sign that there's potential brain damage. They just don't know how bad or how extensive, but that's usually a bad sign. When they see that, then it's like, we need to get you some help quickly. So so you use your reflexes to basically determine how functional your physiology is, right? And so there's a lot of them. I'm not going to go through all of them by any stretch of the imagination, but they're pretty much all the same, right? They're all just basically hardwired in there, typically for purposes of survival. The other thing that I think is always kind of cool, this is just gee whiz sort of stuff that I like, is when you're looking at the spinal nerves as they sort of go out and sort of innervate the body. You can see the cervical area, you can see the thoracic nerves, the lumbar nerves, and the sacral and coccygeal nerves. But the cool thing about this is you can actually map these spinal nerves out to your body, right? So like, for instance, the blue area is largely controlled by cervical plexus and nerves coming out of the cervical area. In the thoracic, you've got the purple zone. So you can see, you can actually map this to zones in your thoracic area. The red, of course, is lumbar, and of course, the sacral is your booty cheeks. Right, that's got to turn around for that one, right? So that's the kind of backside, which makes a lot of sense, right? Because that's kind of where a lot of the musculature and stuff like that is associated with the coccyx and things of that nature. But this is kind of cool, right? Because if you're at all savvy about like paralysis or anything like that, depending on where the nerve damage is happening at which particular point, you can actually pinpoint roughly about where it is based on where the sensation ceases, right? So like, for instance, if you're talking about paralysis and you can't feel anything from the nipples down, then you're talking about some sort of a compromise up here in the cervical and upper thoracic area, right? That's bad news. So like if you're a quadriplegic, then you're talking about usually damage in the cervical region, which then gives you paralysis in all four limbs, because everything from here down becomes responsive. If it's more lower in your spinal cord, like if it's a lower back injury, then typically you will usually be paraplegic. So you'll lose sensation and ability to move from here down, but your upper region will still be largely well innervated. And so you can actually, a lot of times when you take a look at different zones, you can actually feel or sense like about where that cutoff is, and you can roughly figure out the ballpark of where that break is. Jamie. It depends. It depends on whether or not it's a spinal nerve damage or if it's the actual core damage. And core damage can be different, right? Because you can have different amounts of depth of core damage. So you can have core damage that is affecting certain of those tracks, so ascending and descending tracks. So you'll have some compromise in your ability to sense stuff or to send things out. But it depends on what the nature of the damage is and where exactly it is on the spinal cord, which tracks are damaged or compromised. And whether or not you're able to send the ascending messages or the descending messages. So that would be a spinal cord thing, right? So if you have global spinal cord damage in this particular case, you're talking about complete paralysis. If you have partial paralysis, then you could imagine that you still have some function of the spinal cord, but you have some paralysis or some damage to some of those tracks. The other one is if you damage the spinal nerve and not the cord itself, then you can get bilateral asymmetric paralysis. For instance, if the damage comes and you have a damage of your spinal nerve or your spinal plexus on this side, then you can imagine you get some paralysis on this side because it's a descendant of that particular side of that spinal nerve. Whereas this one is fine because it didn't get damaged on this side. That's if you have like some sort of a weird angular blow in this particular case. Yeah. So like if you take, you can really, this is just a general map. So you can really get specific with this map. And depending on the nature of how that damage works and what particular communication segments of the actual nervous system you've damaged will create these different sort of spectrums of where the actual paralysis is located. So you could have like full paralysis in some areas because of the extent of the damage, where you may have partial paralysis in others. So you could have graded paralysis depending on the nature of the damage, but it's all basically mapped back to what the nature of that damage actually is. Yeah, if your phrenic nerve is offline, then you're dead. Which is the reason why the cervical breaks are among the most problematic. And usually what happens is somebody who has a cervical break, they're oftentimes they're really battling and they're struggling with ongoing chronic struggles because of the nature of the things that are controlled by that cervical area. Because you do, so we were just talking about spinal stuff, but you do have cranial nerves that come through that particular area as well. And there's a lot of ongoing issues depending on what you're looking at. So when you're looking at spinal cord injuries, the further up you go in terms of the site of injury, the worse it gets. And yeah, if you do compromise it to the point where you damage or you compromise the signal that you're sending out to the phrenic nerve, then you're dead. Yeah, it's not functioning, that's true. But all that is not governed by spinal nerves. That's not spinal nerves, it's by cranial nerves, right? So most of your visceral core systems are actually governed by cranial nerves. And that's those 12, right? The Roman numerals. And I'll kind of sketch those guys out along with some of the brain areas. And the one thing I want to really talk about is the parasympathetic and the sympathetic innervations, right? Those two I want to make sure we talk about, and just sketching out a little bit of light brain. There's a lot going on in the brain. I can't cover it all. That's the next chapter. But just for the spinal cord, it's pretty basic. Once you find those dorsal roots, then you start to break out into these different sort of plexus structures where they just kind of wrap around your body. And so this is just kind of an example of where these plexus come from. So What I want to do next time is focus on the brain and the cranial nerves and the parasympathetic and sympathetic. Okay. Quick question, and I know we're kind of a little late in going, but the cranial nerves are obviously on the lab exam, right? So hopefully I will, the PowerPoint is posted online. And if you want a really, really good study of the cranial nerves, which is essentially what I would do because we're kind of out of time. But if you go to chapter 13 and you download this, they actually have a really, really nice layout of the cranial nerves that I will do on Tuesday. And obviously, because of the nature of how we do exams, that'll be post exam. So I know it's not going to help you for the exam, but I just want to draw your attention to this because this is basically a really nice breakdown that Seeley does of the different cranial nerves, just going 1 to 2 to 3, and it gives you a really nice view of what they're doing, how they're mapping to the brain. Okay, so I would, if I were you, add this to your study list as you're preparing for the lab exam. I would use this very strongly to study because it does a great job of fleshing out these cranial nerves, all 12 of them, and sort of defining what they do. Tuesday is going to be lab exam like usual. And after you guys are done, then we're going to do our cherry picking through chapter 13, working on brain, the cranial nerves, and the autonomic nervous system. Those are the big pieces, right? The pieces that you need to have in place for AMP2. Okay. And then all of that up to that point by the end of Tuesday is going to be all essentially the last lecture exam material. For the lecture exam? Well, part of 9, right? Because there was a little bit of 9, the musculature, the last piece of musculature when we talked about, wave summation and TREP and incomplete tetanus and all that sort of stuff is going to be on there forward. Yep. Including the neuromuscular junction stuff, right? Because that was basically neurology from a muscle perspective. And that'll all be there as well. Nervous system stuff. And the models, basically, in the models for the lab exam. Yeah. Okay. Yep. Okay.